Hey everyone, and welcome back for another deep dive. You ready to explore some fascinating bacteria today?
Absolutely. I'm always up for a good bacterial adventure.
All right, so today we are going deep into the world of treponemes.
Ooh, treponemes.
Yeah, those spiral-shaped bacteria that just refuse to go away.
You'd think with penicillin and all our public health efforts, we'd have them under control by now.
Right, you'd think that, but
But they're still out there causing trouble all over the world.
that's why we're doing this deep dive. We're going beyond the basics and really dissecting what makes these organisms tick and why they continue to pose a challenge even for seasoned medical microbiologists.
And we'll be using principles and practice of clinical bacteriology as our guide.
So if you have that handy, grab it and follow along.
Let's dive in.
All right, let's start with the star of the show, Treponema pallidum, the culprit behind syphilis. This bacterium is infamous for its unique corkscrew shape.
Yeah, we're talking 6 to 14 tight spirals all packed into this tiny little bacterium. It's amazing, really.
It really is.
It is.
And that unique shape actually makes it incredibly difficult to see under a normal microscope.
Oh, absolutely. It's so slender, just a fraction of a micrometer in diameter. You practically need a special microscope just to get a good look at it.
It's like trying to find a microscopic needle in a haystack.
Exactly.
And speaking of surprises, I was reading that T. pallidum has this really unusual outer membrane. It looks like a gram-negative bacteria at first glance.
Right. It does.
But then you realize it's missing a key component.
It's missing lipopolysaccharide, or LPS. That's a hallmark of your typical gram-negative bacteria, and it plays a huge role in triggering our immune response. But T. pallidum doesn't have it.
So no LPS. That's like a burglar disabling the alarm system before breaking into a house.
Yeah, that's a great analogy. And this lack of LPS could explain why T. pallidum is such a stealthy invader.
It just slips past our immune system undetected, making it even harder to fight off.
Right.
So we've got this incredibly thin, spiral-shaped bacterium that's practically invisible under a normal microscope and has evolved to evade our immune defenses. What else makes this bug so tricky?
Well, its slow generation time is another factor. T. pallidum takes its sweet time to replicate. We're talking a generation time of about 30 to 33 hours.
So it's playing the long game, huh?
Exactly,
Right.
slowly but surely establishing itself.
And this slow generation time has some pretty big implications for both laboratory diagnosis and treatment.
Oh, it does.
Makes it very difficult to culture.
It's very difficult.
Before we get ahead of ourselves, let's not forget about T. pallidum's lesser-known relatives.
Oh, you mean the other treponemes that cause those endemic treponematoses?
Exactly. We've got Treponema pallidum subspecies, endemicum causing beetle, Treponema pallidum subspecies pertany behind yaws, and Treponema coradium, the culprit of pinta,
A whole family of troublemakers.
each with their own signature moves. All right, and now that we have a handle on who these bacteria are, let's get to the juicy stuff. How do these tiny organisms wreak so much havoc in the human body?
Ah, pathogenicity. This is where T. pallidum really shines as a master invader. It enters our bodies through tiny breaches in the skin, or mucous membranes, often so small we don't even notice them.
So we're talking cuts, scrapes, even microscopic tears.
Precisely. And what's remarkable is how few organisms are actually needed to establish an infection.
Okay, now I'm really intrigued.
Yeah.
How many were we talking about?
Studies have shown that as few as 10 organisms can be enough to trigger a full-blown infection.
Wow, 10. That's incredible. I always assumed it would take a massive onslaught of bacteria to overwhelm our defenses.
It really highlights just how efficient these bacteria are.
It makes you think twice about every little paper cut.
Right.
So once these few bacteria gain entry, they waste no time spreading throughout the body. They have this incredible ability to attach to host cells and even squeeze between tissues, evading our immune system's patrols.
They're like tiny ninjas.
And there's this theory that T. pallidum might actually be a stealth organism.
Oh, right. The idea is that it actively avoids detection by our immune system, maybe by cloaking itself in host proteins or by suppressing the immune response. We're still not entirely sure how it does it, but it's a fascinating area of research.
So we have this incredibly sneaky bacterium able to slip in through the tiniest cracks, spread rapidly, and potentially even disguise itself from our defenses. It's a recipe for a silent invasion. But what happens once this invasion takes hold? What are the clinical consequences for the unfortunate host?
That's a great question, and it leads us to the clinical features of syphilis, which are as diverse as they are fascinating.
Okay, let's break this down because I know some people might be thinking syphilis. That's easy chancre rash game over.
Oh, but it's not that simple.
Not at all. Syphilis is notorious for its varied presentations, often mimicking other diseases.
It's a real chameleon.
It progresses through these distinct stages, each with its own set of clinical features. So walk us through these stages. What should medical microbiologists be on the lookout for?
Well, first we have primary syphilis.
Right.
Typically, this is marked by the appearance of a chancre.
Ah, yes, the classic chancre.
It's a painless ulcer with a hard base, usually appearing at the site of infection about 3 weeks after exposure.
Now, these chancres can be pretty sneaky, right?
Well, they can be. They can pop up in all sorts of unexpected places.
Right.
And sometimes people don't even notice them.
Especially if they're not causing any pain.
Right. And to make matters worse, they often heal on their own, even without treatment.
So people might think they're in the clear,
But the infection is far from over.
that brings us to secondary syphilis, which can manifest weeks or even months after that chancre disappears.
And this is when things can get a bit more dramatic.
So what happens during secondary syphilis?
Well, it's often characterized by a widespread rash, which is pretty distinctive because it often involves the palms of the hands and the soles of the feet.
Okay, so that's a big red flag right there.
It is. You can also see fever, swollen lymph nodes, fatigue, sore throat, headache, muscle aches. It can really mimic a lot of other conditions.
So diagnosing secondary syphilis can be a real challenge if clinicians aren't considering it in the differential diagnosis.
Yes. Absolutely. And to make things even trickier, these symptoms can come and go, making it even harder to pin down.
Wow, T. pallidum really is playing hide and seek with our immune systems.
Yeah.
And with us trying to diagnose it.
You could say that.
But what happens next? Does it just keep flaring up like that?
Not necessarily. After secondary syphilis, the infection enters a latent phase where there are no noticeable symptoms at all.
So it goes underground.
Exactly. But here's the catch. Even though there are no symptoms, the bacteria are still present in the body, silently lurking and potentially causing damage.
It's a ticking time bomb. And how long can this latent stage last?
It can last for years, even decades. In fact, about 2/3 of individuals with untreated syphilis will remain in this latent phase for the rest of their lives.
So they might never experience another symptom, but they're still carrying the infection and could potentially transmit it to others.
That's right. And unfortunately, for about 1/3 of those with untreated latent syphilis, the infection will eventually progress to the most serious stage, tertiary syphilis.
Okay, now we're talking about the really scary stuff. What kind of damage are we talking about here?
Tertiary syphilis can affect virtually any organ system, leading to a wide range of complications. We're talking about cardiovascular syphilis with inflammation of the aorta, potentially leading to aneurysms. We're talking about neurosyphilis affecting the brain and spinal cord, causing dementia paralysis and other neurological problems. And we're talking about gummatous syphilis, where these destructive tumor-like lesions called gummas can develop in various tissues.
This is a stark reminder that even though we have effective treatments, syphilis is not something to be taken lightly. Early detection and treatment are absolutely crucial to prevent these devastating long-term consequences.
I couldn't do more. And speaking of detection, let's shift gears and delve into the complexities of laboratory diagnosis.
All right, let's head into the lab. Now, we know that directly detecting T. pallidum is not exactly a walk in the park.
Oh, no, it's not.
These bacteria are notoriously difficult to grow in culture thanks to their slow generation time and their very fastidious nature. So what tools do we have in our arsenal?
Well, one of the classic methods is dark field microscopy, which uses special illumination techniques to visualize those incredibly thin spirochetes against a dark background.
So it's like looking for fireflies on a moonless night.
Yeah, you need just the right conditions to spot them.
Exactly. And while dark field microscopy is considered the gold standard for direct detection, it's not always feasible or available in every lab.
Right.
So what are some other options?
Well, that's where immunofluorescence staining comes in.
Okay, immunofluorescence. I remember that from my microlab days. You're basically tagging the bacteria with fluorescent antibodies, right?
That's right. We use antibodies specific to T. pallidum that are conjugated to fluorescent dyes. When these antibodies bind to the bacteria, they light up under a fluorescence microscope, making them much easier to see.
So it's like giving those tiny bacteria a neon highlight so we can't miss them.
Exactly.
But what about those newer molecular methods like PCR? I know PCR has revolutionized the diagnosis of so many infectious diseases.
It has.
Surely it's being used for syphilis as well.
Oh, absolutely. PCR has become increasingly important for detecting T. pallidum DNA in various clinical specimens, especially in those tricky cases where direct visualization is difficult or when we suspect neurosyphilis.
Neurosyphilis. That's when the infection spreads to the brain and spinal cord, right?
Mm-hmm.
And I imagine it's not always easy to diagnose.
It's not, because getting a sample of cerebrospinal fluid is a bit more invasive than swabbing a lesion.
Right.
But PCR allows us to detect even tiny amounts of T. pallidum DNA in cerebrospinal fluid.
So it's a really valuable tool for diagnosing neurosyphilis.
It is.
But even PCR has its limitations, right?
It does. It's
Yep.
still not as widely available as other diagnostic methods, and there are some challenges with standardizing protocols and ensuring accuracy and reliability across different labs. Plus, it can be more expensive than traditional serological tests.
Okay, so we've got the direct visualization methods like dark field microscopy and immunofluorescence, and we have the power of molecular detection with PCR.
Right.
But I know there's another major player in syphilis diagnosis.
You're talking about serological tests?
Exactly. Serological tests. They're widely available, relatively inexpensive, and can provide valuable information about both the presence and stage of infection.
They really are the cornerstone of syphilis diagnosis.
Now, I know this is where it can get a bit tricky for those who aren't steeped in syphilis serology. Can you walk us through the different types of serological tests and what they tell us?
Absolutely. We have two main categories: non-treponemal tests and treponemal tests.
Okay, let's start with the non-treponemal tests. These are often used for screening, right?
They are?
But I'm a little fuzzy on how they actually work.
Well, non-treponemal tests like the VDRL test and the RPR test detect antibodies that our bodies produce in response to the lipids that are released from damaged host cells during infection.
So they're not actually detecting antibodies against T. pallidum itself, but rather against the collateral damage caused by the infection.
You got it. And that's why they can sometimes give us false positive results. These antibodies can also be produced in other conditions that cause similar kinds of cellular damage.
So that's why we need to confirm any positive non-treponemal tests with a more specific test.
Exactly. And that's where treponemal tests come in.
Okay, so the treponemal tests, these are like the confirmation crew.
Yes.
Coming in to verify the identity of the culprit.
Precisely. Tests like the TPHA and the FTA-ABS actually detect antibodies that specifically target T. pallidum antigens.
So they're highly specific for syphilis.
They are.
But the downside is that they don't really tell us much about the stage of the infection or whether the infection is active or has been treated.
That's right. So we often use both types of tests together.
The non-treponemal tests to kind of assess the activity of the infection and monitor the response to treatment.
Because their titers tend to decline with successful treatment.
And the treponemal test to confirm the diagnosis.
Exactly. It's a team effort.
And while we've made some pretty significant progress in syphilis diagnostics, some challenges still remain.
They do.
Like confirming neurosyphilis.
Yes, neurosyphilis is still tricky.
We don't have a perfect method yet for directly detecting intrathecal antibody production.
Right, which would definitively tell us if the infection has reached the central nervous system.
So there's always room for improvement. But let's zoom out a bit and talk about the big picture epidemiology.
Yeah.
Where do these treponemal infections fit into the global landscape of infectious diseases?
Well, syphilis and endemic treponematoses are still significant public health concerns worldwide.
They are.
And unfortunately, we've seen a resurgence of syphilis in recent years, both in developing countries and in industrialized nations.
That's concerning. What's driving this resurgence?
Well, there are a number of factors at play. Changes in sexual behavior, including an increase in unprotected sex, have definitely contributed to the spread. And we're also seeing a rise in syphilis among men who have sex with men.
And the presence of HIV co-infection can also complicate both diagnosis and treatment.
Absolutely. And of course, social and economic factors like poverty and lack of access to health care also play a role, especially in those endemic treponematoses.
It's a complex interplay of factors. And speaking of complexity, let's talk about prevention and control. What strategies are being employed to combat these tenacious treponemal infections?
Well, when it comes to syphilis, the classic public health strategies are still our best bet, promoting safer sex practices, ensuring access to testing and treatment, and conducting effective partner notification and contact tracing.
So it's all about education awareness and breaking down those barriers to testing and treatment.
Exactly. But it's not always easy, especially in those outbreaks we were talking about where anonymous sexual encounters make traditional partner notification nearly impossible.
Right. You can't track down and treat someone if you don't know who they are.
Exactly. So public health experts are exploring some new approaches, things like using the internet and social media for partner notification, targeting screening programs in venues where transmission is more likely to occur, and even considering the feasibility of mass treatment strategies.
So thinking outside the box and adapting to the changing landscape of sexual networks.
Precisely. And when it comes to those endemic treponematoses, control measures often involve more community-based approaches, things like mass drug administration campaigns, active case finding, and health education initiatives.
It's all about meeting people where they are and empowering them with knowledge and access to care. But let's be honest, all the prevention and control strategies in the world aren't going to eliminate these infections entirely. So let's talk about treatment. What's the magic bullet for these tricky treponemes?
Well, the good news is we do have a highly effective treatment, penicillin.
Penicillin, the classic wonder drug.
It's been the gold standard for treating syphilis and other treponemal infections for decades.
But I imagine the type of penicillin and the duration of treatment can vary depending on the specific infection and stage of disease.
Oh, you're absolutely right. For primary, secondary, and early latent syphilis, a single intramuscular injection of benzathine penicillin G is usually sufficient. But for latent syphilis, neurosyphilis, or syphilis during pregnancy, we typically use multiple doses of penicillin administered over a longer period.
So it's not a one-size-fits-all approach. We need to tailor the treatment to the individual patient and their specific circumstances.
Exactly. And of course, we need to consider things like potential allergies to penicillin and how to manage treatment in special populations like pregnant women and HIV-positive individuals.
Right. It's all about personalized medicine, understanding the nuances of each case. But what happens after treatment? Is that the end of the story?
Not quite. Management of infection involves more than just giving someone antibiotics. We also need to make sure the infection is truly eradicated and that we prevent reinfection or transmission to others.
So what does that look like in practice? What are the key steps in managing a patient after treatment for a treponemal infection?
Well, one of the most important aspects is follow-up. We need to monitor patients very closely after treatment, checking for any signs of relapse or reinfection.
So we're basically making sure those bacterial ninjas haven't gone back into hiding.
That's a great way to put it.
And another critical aspect of post-treatment management is partner notification.
Absolutely. We need to track down anyone who might have been exposed and make sure they get tested and treated as well.
And this can be a real challenge, especially when dealing with sensitive or confidential information.
It can. Partner notification requires sensitivity, discretion, and strong communication skills. It's about building trust and making sure that everyone involved understands the importance of testing and treatment.
It's a delicate balance, but it's absolutely essential for preventing the spread of these infections.
Precisely. So as you can see, managing treponemal infections is a multifaceted process that requires a combination of clinical expertise, laboratory skills, and public health awareness.
Wow, we've covered a lot of ground already in this deep dive. We've explored the unique characteristics of these spiral-shaped bacteria, their stealthy ways of causing infection, the complexities of laboratory diagnosis, the challenges of epidemiology and control, and the importance of personalized treatment and follow-up.
And we're just getting started.
But I think it's time to put your knowledge to the test. Are you ready for some exam questions?
Hit me with them.
All right, let's see what you've learned. Here's your first challenge.
What are the key differences between the outer membrane of T. pallidum and that of typical gram-negative bacteria?
Think carefully about those structural nuances we talked about earlier. Okay, question number 2.
Describe the 4 stages of acquired syphilis and their characteristic clinical features.
Remember, syphilis is a master of disguise, so pay attention to those subtle clues. All right, question number 3.
What are the advantages and limitations of using non-treponemal and treponemal tests for diagnosing syphilis?
Think about those different testing strategies and how they work together. Moving on to question number 4.
Why is it so difficult to control recent syphilis outbreaks, particularly among men who have sex with men?
Consider those social and epidemiological factors we discussed. And finally, your last challenge for this part of the deep dive.
Explain the role of molecular typing techniques in epidemiological studies of syphilis.
All right, take a moment to ponder those questions. We'll be back shortly to discuss the answers in part 2 of this deep dive into the world of Treponemes.
Looking forward to it. Welcome back, everyone. Did you have a chance to ponder those exam questions?
I hope you didn't find them too tricky.
Let's dive into the answers and see how you did.
All right, let's start with that first question about the outer membrane of T. pallidum. What are those key differences between this bacterium and your typical gram-negative bacteria?
Right. So the big difference really boils down to that missing lipopolysaccharide, or LPS. Remember, LPS is a major player in most gram-negative bacteria. It's like a giant, hey, look at me Sign for our immune system.
But T. pallidum's like, nope, not playing that game.
Exactly. It ditches the LPS and slips right past our immune defenses.
A master of disguise.
It really is. And this lack of LPS is probably a big reason why T. pallidum is so good at evading our immune system.
It makes it so much harder for our bodies to fight off the infection.
And it also makes it harder to develop a vaccine.
Oh, that's right. Traditional vaccine approaches often target prominent surface molecules like LPS. But if T. pallidum doesn't have that...
We need a new strategy.
Exactly. It's like trying to design a mousetrap for a mouse that doesn't like cheese.
Researchers are definitely working on it, though, looking for alternative ways to trigger that immune response. It's cutting-edge stuff.
It's a reminder that we're in a constant arms race with these bacteria.
Yeah.
They're evolving, and we have to keep up.
Absolutely. All right, ready for question number 2?
Bring it on.
Okay. Describe those 4 stages of acquired syphilis and their characteristic clinical features.
This one's all about remembering those subtle clues that T. pallidum leaves behind as it progresses through the stages of infection.
It's a real detective story.
So let's start at the beginning. Primary syphilis. What's the hallmark sign?
The chancre, that painless ulcer with the hard base, it usually shows up at the site of infection about 3 weeks after exposure.
The classic chancre. But as we discussed earlier, it can be pretty sneaky sometimes.
Oh, it can appear in all sorts of places. And it often heals on its own even without treatment, so people might not even realize they've been infected.
Right. It's a silent but deadly first act.
Then comes secondary syphilis, which is when things can start to get a bit more dramatic.
This is when T. pallidum really starts to make its presence known.
Yeah, we see those widespread rashes, often involving the palms and soles, which is a pretty distinctive feature.
It's a big clue that something's up.
And you might also see fever, swollen lymph nodes, fatigue, sore throat headache, muscle aches. The symptoms can be quite varied and often mimic other conditions, so it can be a real challenge to diagnose.
Especially if clinicians aren't thinking about syphilis as a possibility.
Exactly. But even after all that drama, the infection eventually enters a latent phase where there are no noticeable symptoms at all.
It goes quiet. But that doesn't mean it's gone.
Not at all. The bacteria are still there, lurking in the shadows, potentially causing damage even without any outward signs.
And this latent phase can last for years. Even decades.
It can, which is why it's so important to catch and treat syphilis early on.
Because if it's not treated, about a third of those with latent syphilis will eventually progress to tertiary syphilis.
And that's when the real trouble starts.
We're talking about serious long-term complications affecting the heart, the brain, the nerves, and other organs.
It's a reminder that syphilis is not something to be taken lightly.
Right, moving on to question number 3. This one's about those 2 main categories of serological tests for syphilis. The non-treponemal tests and the treponemal tests.
What are the advantages and limitations of each?
Right. So let's break it down, starting with those non-treponemal tests like the VDRL and the RPR.
These are our workhorses for screening. They're widely available, relatively inexpensive, and pretty quick to perform.
But they're not without their limitations.
Right. They're not directly detecting antibodies against T. pallidum. They're detecting antibodies against those lipids released from damaged host cells, which means they can sometimes give us false positive results.
So they're sensitive, but not always specific.
Exactly. Which is why we always confirm positive non-treponemal tests with a more specific test, like one of the treponemal tests.
The TBPA, the TBHA, or the FTA-ABS?
Right. These tests directly target those antibodies that are specific to T. pallidum antigens. So they're much more specific for syphilis.
They're like the double check to make sure we're not being fooled.
Exactly. But the trade-off is that they don't tell us much about the stage of the infection or whether it's active or not.
So we use them in conjunction with the non-treponemal test to get a more complete picture.
Exactly. Okay, ready for question number 4.
Hit me.
Why are those recent syphilis outbreaks, particularly among men who have sex with men, proving so difficult to control?
This is a tough one. It's a complex issue with a lot of factors at play.
One of the biggest challenges is the anonymity factor.
Right. With a lot of anonymous sexual partners, traditional contact tracing becomes nearly impossible.
It does. You can't track down and treat someone if you don't know who they are.
And we also have to consider changes in sexual behavior, including increased unprotected sex, which obviously increases the risk of transmission.
And then there's the added complication of HIV co-infection, which can affect both the course of the disease and the response to treatment.
It's a reminder that these infections don't exist in isolation. They're intertwined with social behaviors and other health factors.
Public health officials are definitely working hard to develop new strategies to address these outbreaks.
Thinking outside the box, coming up with creative solutions.
Exactly. All right, last question for this part.
Bring on the final challenge.
Explain the role of molecular typing techniques in epidemiological studies of syphilis.
This one's all about those microscopic fingerprints that help us track these bacterial invaders.
Molecular typing allows us to analyze the DNA of different T. pallidum strains and figure out how they're related to each other.
So we can see if outbreaks in different locations are caused by the same strain or by different strains.
Exactly. And that can help us understand how the bacteria are spreading within a population and even pinpoint the sources of infection.
It's like having a GPS tracker for bacteria.
It really is, and it's a powerful tool for understanding the epidemiology of syphilis.
So there you have it, the answers to our syphilis exam questions.
How did you do?
I hope you learned something new today about these fascinating and challenging bacteria. Molecular typing really is a game changer. It's helping us understand the spread of syphilis in ways we never could before.
Absolutely. It's like having a secret window into the world of these bacteria.
But even with all these amazing advancements in technology, we can't forget about those basic public health measures.
Oh, absolutely not. Those are still our foundation for controlling these infections.
Things like promoting safer sex practices,
Right.
increasing access to testing and treatment,
Yes.
strengthening partner notification programs.
Those are all crucial.
We can have all the fancy diagnostics and treatments in the world, but if people can't access them or aren't even aware of the risks...
It's all for nothing.
Exactly. It's
Yeah.
about connecting the dots between the lab and the community.
And empowering people to take control of their health.
Well, I think this deep dive has given our listeners a lot to think about. We've explored the world of treponemes, from their unique structure to their sneaky ways of causing infection,
We've talked about the challenges of diagnosis
the complexities of epidemiology and control.
and the importance of a holistic approach to treatment and management.
These bacteria are truly fascinating.
They are. They've evolved all these strategies to evade our immune systems and spread silently within our bodies.
But we're learning more and more about them every day.
And developing new tools to fight back.
With ongoing research and a strong commitment to public health.
We can make a real difference.
So as you head back to your labs and clinics, remember what we've talked about today.
Stay curious, keep learning.
And be vigilant.
Early detection is key.
Absolutely. Thank you for joining us on this deep dive into the world of treponemes.
It's been a pleasure.
We'll catch you next time for another exciting adventure in the microbial world. Until then, stay safe and keep those microbes in check.